All right, in this lecture, I am going to cover section 8.5, the light-dependent reactions, basically looking at specifically plants with their two photosystems. So the last time we lectured on what a photosystem is, what makes up a photosystem. A photosystem, remember, is a series of pigments like chlorophyll A and chlorophyll B and some accessory pigments, and they're kind of clustered together on an antenna complex. And when a photon hits it, we have that photoelectric effect, where electrons get excited, they gain energy, and they get excited and reach a higher energy level. And so we are going to elaborate what light-dependent reactions do. You guys did the POGL packet, which is going to be due, I think, tomorrow. Yeah, tomorrow. And so you saw some of this, and so we'll just kind of go into depth of what the light-dependent reactions are. So the photo part of photosynthesis. So remember that some bacteria, they just have a single photosystem, and that means that things get recycled back at the end. But plants use a two photosystem process, and they are linked together. Okay, so it looks like this Z, and it's non-cyclic. Okay, whereas with some bacteria, when they just have the single, then it is cyclic. So there are two connected photosystems here, and they are linked to overcome the cyclic photophosphorylation that occurs in the bacteria where they recycle electrons. So here we have an alternative source of electrons from the oxidation of water. Water is going to split, and when water splits, we generate oxygen, okay, and as well as two protons, and then the electrons are the ones that replenish the photosystem, those electrons that get excited. Non-cyclic transfer produces an electron carrier called NADPH. So here are the two photosystems in brief. I will abbreviate them as PS1 and PS2. So PS1 has an absorption peak of 700 nanometers, or P700, and that is the reaction center. And actually, I'm just going to go back here real quick. Okay. All right, I just want to make sure I didn't mess something up there. I didn't. Okay, so that means that reaction center gets excited or responds to wavelength visible light that is 700 nanometers. Okay, so kind of that reddish spectrum. And it's going to transfer electrons to NADP plus to produce NADPH. This photosystem is very similar to the photosystem that is found in sulfur bacteria. Photosystem 2 has an absorption peak of 680 nanometers, okay? And this has higher energy because, remember, with wavelengths, this is higher. It generates enough potential to oxidize water, where water will split. And when water splits, it replaces those electrons that get transferred to photosystem 1. So they work together, they transfer electrons, and what they do produce is ATP and NADPH, which will go on to the light-independent side of photosynthesis. So here, yes, they work together. So let's talk about how do we know that these two photosystems work together in non-cyclic photophosphorylation. It comes from the rates of production based on red and far red light. So they did something called an enhancement effect, where they exposed plants to like one type of light, and they measured the output. And then they looked at the far red light and looked at the output, and they realized that when you just exposed them to one or the other, the production rate wasn't as high as it should be. And they're like, what's going on here? So then they exposed the chloroplasts to both lights at the same time for this enhancement effect. And realize that there's two photosystems at play here, one that prefers red and the other in the far red light. So here's the enhancement effect graph where, hey, the photosynthesis rate is not where it should be. Here's the red with the 680, and then we put that together, and now you're getting the rates that you should see. Okay, so remember in plants, it's non-cyclic phosphorylation. The electron paths are not circular. They do get ejected, and they end up with an electron carrier called NADPH. The path is like a Z, so we're going to go through the Z here. Photosystem 2, the high energy electrons are used to make ATP, and they get passed on to photosystem 1, and then photosystem 1 actually produces the NADPH. Now, this is going to be super, super confusing, but the photosystems, they're backwards, okay? So PS2 happens first, and then PS1, and that's because PS1, the photosystem, was first discovered, and then they realized that there was a photosystem in front of it, and they couldn't like really rename it. So photosystem 1 occurs second, and photosystem 2 occurs first. So this would be photosystem 1, and this would be photosystem 2. Or sorry, oh gosh darn it, I just messed myself up. This is photosystem 2, and this is photosystem 1. Yeah, don't do a lecture when you're sick. Okay, so here we have water, and with photosystem 2, at the 680, P equals 680, it's just enough energy to oxidize water and split it into oxygen and 2 protons, okay? And these protons will play a role later on in the ETC. But those electrons get excited, and they're going to get passed through the ETC, pumping hydrogens across, and then it will reach photosystem 1 with the P700. Wow. From photosystem 2, we generate ATP, and photosystem 1 produces NADPH. So photosystem 2, we have an antenna complex, we have chlorophyll A and accessory pigments bound to protein channels. That reaction center is the P680, and what's really at play is these 4 manganese atoms that are essential to oxidize water. So this is a really rudimentary picture of photosystem 2, but you can see the 4 manganese, water comes in, and it gets oxidized to produce oxygen and 4 protons. So the 4 manganese atoms bond around the reaction center, and then 2 water molecules will bind to them. So yeah, we're basically removing those electrons from water, and then they head to the manganese, and then those electrons get removed right there. So the water, the electrons from water are replenishing the ones that get lost from that photoelectric effect. So you need 4. They have actually quantized how many photons are needed. 4 photons are needed to complete the oxidation of 2 water molecules, and as a result, you produce 1 oxygen in the process. And that's why you see half an oxygen molecule sometimes in this equation. Okay, if they just have 1 water molecule, you might see half oxygen. So this is 4 photons plus 2 H2O, and that should be your arrow. I'm doing this in Google Slides because my MacBook updated, and I can't open up PowerPoints. So yay, it's been a great day. Okay, so after those electrons get excited, they're going to go to a higher energy level, and they are going to get picked up by an electron acceptor called a quinone, or PQ. Yes, PQ. So reduced means gain. So blastoquinone, PQ. That's very similar to the molecule in mitochondria. PQ will donate it to a proton pump called B6F, which is in the thylakoid membrane. And then after it reaches or passes B6F, then a different electron acceptor will pick it up, plastocyanin, PC. This contains copper. So then we reach photosystem 1, where the reaction center is P700. Okay, energy gets fed through the antenna complex. We do have some chlorophyll A as well as some accessory pigment molecules, but photosystem 1 will accept those electrons from PQ. Then these electrons will get re-energized. Okay, they absorb some more photons from the pigments up here, and they reach a very high energy level where they will get passed on to a non-sulfur protein called ferredoxin. Then ferredoxin will get reduced because it picks up those electrons. We are now on the stromal side of the membrane, and it will carry those electrons and donate it to NADP plus to become NADPH through an enzyme called NADP reductase. So essentially this is the diagram here. Photons come in, bounce around the chlorophyll A and accessory pigments till they reach the reaction center of P680. We have our manganese, okay, with the photoelectric effect, enough energy to split water to produce 2 hydrogen ions or 2 protons, and at oxygen molecule there's that half because we just have one water molecule. If I had two, then I would produce a full oxygen molecule. But those electrons get replenished in the reaction center. So they get a higher energy level where it gets passed on to PQ, okay? So you don't need to worry about all the, I don't know what the hell that is, but PQ will carry to the B6F complex, and this is a proton pump, pumping hydrogen across. Then it will get picked up by PC, and then carried to photosystem 1 with the P700 reaction center, where those electrons get even more energized, higher than photosystem 2. And then we are going to hit ferredoxin. And this one doesn't show the NADP reductase. I'm not seeing it on here, but NADP plus a proton and an electron will become NADPH. Now these protons that are going to pumped across, we're going to build up that concentration gradient. It will flow through ATP synthase, generating that ATP. And both ATP and NADPH will be used in the light-independent reactions, aka Kelvin cycle. So ATP is generated by chemiosmosis, very similar to cellular respiration. The proton pump that's responsible for building that concentration gradient is that B6F complex. So yeah, they get pumped from the stroma to the thylakoid compartment. So just know that that gradient's building up to the thylakoid compartment. If you take a look at the thylakoid structure, you can definitely see just how this all kind of works. So there are 4 complexes responsible for light-dependent reactions. We have PS1, PS2, we have the B6F complex, and then we have ATP synthase. And they are not randomly arranged in the thylakoid membrane. They are arranged in a way where one gets passed on to the other to increase efficiency. So like PS2 is found in grana, and PS1 and ATP synthase are found in the stroma. It's supposed to be stroma lamella, or the edges of grana. And then we have our cytochrome B6F complex between the grana and the stroma. I'm sorry. So here's photosystem 2, photosystem 1, cytochrome B6F, and ATP synthase. So they are arranged in a way where they can pump things across and build up concentration gradients and flow through ATP synthase. So, okay, so that's it for notes. You will take a quiz on light-dependent reactions, basically PS2 and PS1, on Wednesday. I will post information in Google Classroom. Your POGO packet will be due tomorrow. I'm going to hand out the guided notes so you can just get a head start on that. So, yeah, that's all I have for today. Tomorrow, I will move on, I think, with the Kelvin cycle. And then we'll probably take the Kelvin cycle quiz Thursday. And then we'll game plan for a lab. Maybe we'll start it. I don't know. We'll figure it out. But hopefully we'll see you tomorrow.